Hello everyone. In this video, we'll see about growth and nutrition of bacteria. Basically, we see about bacterial growth curve and the factors which influence the growth of bacteria. Okay. So, what is generation time? So, generation time or the doubling time is the time taken by the bacterium to divide into two daughter cell. Okay. So, division of bacteria takes place by binary fission. So cell division. Okay, so one bacteria will divide and forms a daughter cell, that is one bacteria become two. Okay, the time taken by the bacteria to double its number, we call it as generation time or doubling time. Okay, so most medically important bacteria, is it at least 20 minutes it will take to double its number. And coming to Mycobacterium leprae, it is 20 days, and Mycobacterium tuberculosis will take 20 hours to double its time. Okay, so this is a bacterial growth curve most commonly asked in the examination. Okay, so bacterial growth curve has mainly four steps, that is lag phase, log phase or exponential phase, stationary phase and decline phase. Okay, so this graph shows four different phases of bacterial growth curve against the time frame. Okay, as the time increases, number of cells will increases in the exponential phase, and slowly it will decline in the death phase. One by one we'll see each and every step. Okay, so when we seed the bacterium or when we inoculate the bacterium to a particular suitable liquid medium, so it will start its growth. Okay, first it will take time to adjust that particular environment. Okay, so lag phase we call it as immediately after seeding or inoculating bacteria into the medium, so there will not be a appreciable increase in the number. Okay, so it will take time to increase the size of the cell, or it will produce enzymes and it will adjust to that particular environment. Okay, so the bacteria needs time to adjust for that particular new environment. Okay, so this phase we call it as lag phase. Okay, this phase will depends upon the bacterial species. Okay, which species you have inoculated and how much inoculum means how much bacteria you have added to that particular liquid media and what nutrient you have provided. So these all factors will depend on the lag phase. Okay, so lag phase basically is a lag means the bacteria will take its own time to adjust for the environment. Okay, here there is no increase in the number, only the shape or the size of the cell will be increased. Okay, in the next phase, that is log phase or logarithmic phase or exponential phase we call. Okay, so in this the actual multiplication of the bacteria will start. Okay, so it will multiply and it will increases its number. Okay, so this is the most crucial phase. Okay, and we have stationary phase. So stationary phase means slowly the nutrients gets exhausted. As the number of cells are increasing, whatever nutrients you have provided, it is utilized, right? So now in stationary phase, slowly the nutrients gets exhausted and toxins are produced by the bacteria. Okay, so toxins start accumulating, so the number of new cell is equal to number of dying cell. So there is no increasing or exponential increase in the size or the number. So here slowly it will take a steady. Okay, it will take a flat in the graph. Okay, so in this phase sporulation occur. The spores are produced in the nutrition depletion condition, you know. So here sporulation we can see. And the last phase of decline. Decline means, or we call it as death phase. Here the number of viable bacteria or the number of live bacteria slowly decreases due to the nutrient exhaust. Okay, so here the activity of the toxin product, autolytic enzymes, everything starts, so the number of cells slowly goes down. Okay, so this is all about the bacterial growth curve. So bacterial growth curve importantly has 4 different phases, that is lag phase, exponential phase, stationary phase and decline phase. Okay, so now we will see about factor which influence this growth. Okay, so there are so many factors which influence the growth. So for our growth also there are so many external factors will be applicable, right? So same thing for the bacterial growth also, there are so many external factors which influence. Okay, so we'll see one by one, that is nutrition, which media or what type of nutrition you are providing, at what temperature you are incubating, and oxygen requirement, pH, salinity, that is salt concentration, pressure, light radiation. These are the 7 different factors mainly influence the growth of the bacteria. Okay, coming to the nutrient, so we have to provide the hydrogen ions, carbon, nitrogen, macronutrients, right, minerals, so amino acid, purines, pyrimidines, how we require carbohydrate, proteins and lipids. So bacteria also requires hydrogen, carbon, nitrogen, macromolecules, micromolecules for its development or the metabolism or the growth. Okay, so coming to the temperature, so there are 3 different temperature ranges actually. So we have psychrophilic bacteria, mesophilic bacteria and thermophiles. Okay, so optimum growth or the optimum growth range of temperature is where we see the maximum growth rate. Okay, so usually the pathogenic bacteria or medically important bacteria are mesophilic, which grows between the temperature of 30 to 37 degree. Okay, psychrophiles means you have the bacteria which grow at 15 to 20 degrees Celsius, between that temperature. Okay, thermophiles are like above 45 or 50 degree it will grow. Okay, coming to the oxygen, this is very important. So obligate aerobes, obligate anaerobes and facultative anaerobes. Okay, so obligate aerobes requires oxygen for their growth. Okay, so bacteria grow only when free oxygen is available. So obligate anaerobes, if you expose to the oxygen it will die. Okay, so bacteria grow in the absence of the oxygen, so we call it as obligate anaerobes. Okay, and we have facultative anaerobe, means bacteria grow in the presence or absence of the oxygen, little amount of anaerobic condition or little amount of aerobic condition, so it will adjust. Okay, so those type of bacteria we call it as facultative anaerobes. So coming to the hydrogen ion concentration, so that means pH level. Okay, so we have neutrophiles, acidophiles and alkalophiles. So you know, right, acidic pH, alkaline pH and neutral pH. So same thing, those grow, the bacteria which grow at neutral pH we call it as neutrophiles. Those which prefer to grow at low pH, that is acidic pH, we call it as acidophiles, and alkalophiles which grow on the alkaline medium. So both uh, acidophile, alkalophile and neutrophile, these are the 3 different types of the hydrogen. Based on the hydrogen and concentration we classify, and most of the pathogenic bacteria grow at best at pH of 6 to 8 range. Okay, so most of them are neutrophile. Okay, so coming to the pressure, osmotic pressure, that is the pressure exhibited by the water molecule or the surrounding media, right? So on the bacterial cell wall. Okay, osmotolerant bacteria can grow solution with a high solute concentration, where the solute concentration is more. Okay, it can tolerate. So that type of bacteria we call it as osmotolerant, and those which which fear or which does not grow that much in the osmotolerant um, environment, so we call it as osmophilic bacteria. And the hydrostatic pressure, so water column we say, right? So we add water to the media preparation, right? While preparing the media. So that water column that exhibits some pressure on the bacterial cell, right? So that we call it as hydrostatic pressure. So we have barotolerant bacteria grow at high hydrostatic pressure, and barophilic which grow at best at high hydrostatic pressure. Okay. So barotolerant and barophilic bacteria. Coming to the light radiation, so we have photosynthetic bacteria, photosynthesis, right? So we see in the plant. So that mechanism some bacteria shows, so which provide or which prepares its food, right? So that are photosynthetic bacteria which requires light in the visible spectrum to carry out the photosynthesis. So there are 7 factors we saw, right? So based on the requirement or the based on the different conditions, the bacteria will grow. So these are the 7 different points or the 7 different factors which influence the growth of the bacteria. Okay, so if you have any doubt, you can comment in the below comment section, so I'll be happy to answer your questions. Thank you.